the Francis Crick Institute, which is one of the institutes that MRC is funding. And my lab, as you heard, is working on cancer. And before digging into the topic of cancer, I'm going to have a little bit of an introduction first on the basic that you need to be reminded to understand what I'm going to define cancer as, what it is and how it works. And also at the end, we are going to see a little bit more what is the angle that we are taking in the lab to study cancer. Okay, so to start with, I'll present you with what you probably know, which is what we call the building block of life. Is essentially the cells. Every living organism on the planet is essentially made by these basic living things, which is a cell. So the cell is, of course, a very complex structure, but the only thing that I would point out is the fact that it has a nuclei, and inside the nuclei there is the genome, and the genome is essentially made by DNA. And the DNA in the genome of the cell are what control what the cell do. So how does it work in a complex organism like a human body? Where are the cells? So essentially they are so they are the building block of life, as I told you. So if we focus on any of the tissue, for example, we will pick here a breast, because particularly in my lab, we have a strong interest in breast cancer. So this is an organ that we are interested in. So if we look at how the organ is actually built on, then we'll find a lot of tissue. And each of those tissue were made by cells. So if you cut, for example, here, if you imagine this is the epithelial cell of the duct, so this is the functional part of this tissue. If we cut and then we, with a magnifying glass, we look inside, we'll find that it's made by cells which have a particular shape and they form this structure. And the same thing will be true for a blood vessel or also fat tissue. If you magnify, you will find that it's actually made by cells which are filled with fat. And this is true with each of every of our organ. So in this case, for example, we are looking at lung, and the functional part of the lung is the alveolar, where the gas exchange take place. And also there will be, vessel, which bring the blood close to the air, which come inside the alveolar. And again, everything is made by cells, which are which have a particular shape, and they are assembled in a particular way so that they form the structure of the tissue. And one interesting thing that it emerged also in this schematic is the fact that all the cells are going to be very different. They have different shape because they have to fulfill a different function. So there are a set of things that each of those cells will have to do, and together they'll fulfill the function of the tissue and the organ. However, as a reminder, all of those cells will have that basic structure. So all those organelles will be in there, and each of them will have their nuclei and their genome. But here we might ask, well, if the genome is what defines what the organism is going to look like and what defines what each of the cells is going to do, how is it possible that even though it's exactly the same in all the cells are so different from each other? And understanding how that works is very important if we want to understand what cancer is. So we have to take a step back to the very beginning. So when we move from one cell to the creation of the whole organism. So in the beginning, we start from one cell, which is a fertilized egg. And the genome here, it will be derived by the union of the mother DNA and the father DNA, and a new DNA now will be created by the union of these two. So it's now unique, and it will lead to the creation of a unique organism. So to use as a metaphoric definition of the DNA contains all the information which are required to build a particular organism. So we can compare the DNA like if it would be a chemical equivalent of actually a book. So it's the book of life, and in this case, it's the book of the life of this particular organism, which is going to be built by this cell. So this cell will now start dividing, And the important thing to notice is that this cell will be able to access literally all the information in that book. That book is completely open and fully accessible to this cell. We call this cell totipotent because it can form every cell in our body. And initially we'll start dividing, and all those cells are going to be totipotent. On the other hand, when they start moving around and taking their shape and creating an embryo and deciding which kind of tissue they want to be part of, then they will start reducing the things that they are allowed to do. And that reduction will come with the restriction of the information of the book. So we'll end up in a situation where once the baby is formed and all the organs are there, each of the cells in that tissue will be essentially reading just one page and probably even one paragraph of that book. And this will restrict what the cell will do, but also will focus the cell to do a particular function. So let's go back to the original question. What is cancer? If you Google the definition, you find that cancer is a condition where the cells in the body are just uncontrolled growing. So how's that working? It has a lot to do with the book we're talking about. So the genome, this cell, which is now differentiated and it does its function in the body, when it becomes a tumor cell, will undergo a process where it unlocks the information of the book. And now suddenly the cancer cell is a cell which is able again to read all the information like the embryonic cell were able to do. And at that point, this cell does not have the purpose to function in the tissue. It gains a new purpose, which is the purpose that the embryonic cell had in the beginning. So it will start growing. It starts to grow because the intention of this cell is to create something new, like an embryo or in a very abnormal way. So all the cells which are going to be generated, on the other hand, they're not necessarily the same. There's going to be a lot of cells which have the same potential of the tumor cell, but other cells are going to lose a little bit of that potential. So those cells are going to be more vulnerable to whatever treatment we want to do, and also they won't be able necessarily to start a new tumor. So the cells which are really more potent are the ones that we really need to tackle to make the whole structure collapse. And this we can see in an early, so because these are the cells which are driving not only a new tumor growth if the cell disseminate, but also they're responsible to hold up the structure of the tumor. And this we could see also in a study that we did in 2008 when I was still not in London. This was during my time in Switzerland. So here we basically identify the cell which originated, in this case, was a skin tumor, and we could genetically deplete them, and the depletion of those cells, you can see, led to the collapse of the whole tumor structure after an initial growth. And this is, of course, possible to do if the tumor is not very advanced. In an advanced tumor, things are a little bit more complicated. It's more hard to identify those cells which are responsible for the tumor growth, and the cells are so powerful that they can modulate their activity and can change from one status to the other. So it's not very easy to target those cells. Otherwise, of course, we wouldn't have a tumor problem anymore. But what is the drive, this unlocking of the tumorigenic power? Well, if we go on and decide and read more about the definition, we'll find that cancer is a genetic disease. And indeed, is genetic mutation drive this dedifferentiation that I was talking about. So there is a damage in the DNA. The DNA is being repaired, but mistake can be made. And if a mistake is done, then a mutation will occur in particular gene. And those genes which are responsible for controlling the restriction of the information that the cell has access to will be an oncogene, which then unleash mutation there, unleash the whole accessibility of the information. The whole embryonic potential will be available to this cell, which now become a tumor cell. So an image that we can have is that a genetic mutation will hit a cell, that cell will turn into a cancer cell, which will start growing without control. This is partially true. Things are a little bit more complex, and here it comes the angle that we are more focusing on in my lab. Because together with this gain of uncontrolled proliferation and resistance to cell death, the cancer cell have another power along when they gain this embryonic power, which is the power to change their microenvironment. So they can change the tissues around them, and that allow them to grow. So the picture we have is more something like that. So we have a cancer cell that starts, and the surrounding of the tissue completely change. And all these healthy cells, which are not mutated, they will now embark in the tumor program and foster their growth. So the tumor microenvironment is essentially the environment where cancer cells exist. And in fact, I'm going to argue that without that tumor microenvironment, cancer wouldn't exist in the first place. So I'm going to go back to the breast here to show you an example visually of how this is happening. So we have the schematic that I showed you before. Now we have a mammary gland we can cut. This is a real gland. We cut it on this direction. You can see the duct here. And now if we look on top of the slice, the tissue is dead, and we can stain it with color. So you can see all around this structure that you see here in the drawing, and all the cell around it. And this is the beginning of a tumor. So you see that the cells are starting to uncontrollably grow. And if we look at the bigger magnification, you can actually see all the different cells which are growing up. And you see the cell structure is here. This is the nuclei, and the body of the cell is a bit pinkish. So you see all these different cells are growing, and they continue to grow. And what I like to point the attention here is when they grow, they also start to be embedded in a very different environment to what they were the normal cell before. So this cluster of denser nuclei, these are all the cancer cells, and all this pink structure here, they're no cancer cells. These cells are normal, they're just coming from the tissue, and they are supporting the tumor growth. Essentially, a tumor is more or less 50-50 cancer cell, which are non-cancer cell, but they are now diverted into the cancer program. And I'm going to show you a little bit more in detail how this worked from the beginning, showing you a very simple experiment we can do in the lab. We can take the cell from this advanced tumor, we can take a small number of them, and then we can put them in a gel and to keep them together, and then insert them in the fat bed, so in the mammary gland of a mouse. And this cell will now generate an exact replica of the original tumor the cancer cell were coming from. So let's look what happened over time, and particularly what happened in the very beginning. So in the very beginning, we have those cells which are inside this gel, inside the tissue. And what can happen is that the tissue cell will infiltrate inside the plug and are going to meet the cancer cell. So you have this early stage where the tumor cell already started to grow, and the brown cell that you see here, they come from the tissue and they start this engagement with the cancer cell. So we created a situation where this interaction couldn't happen. So now we have the cancer cell, but the tissue cell cannot come in. And if that's the case, the tumor cell in the beginning, they'll grow equally well, but you see they're alone. Nobody else is there. So what happened over time? Well, over time, we can see that the tumor will be formed in the case that the cell have someone to talk to, but in the case where no one is there to talk to, now there's no tumor. And the cancer cell do grow in there. They grow in there in a way that is very similar to what they would have done if they were normal cell in tissue. And I'll show you here as a comparison how a normal mammary gland duct look like. Very similar to this very aggressive cancer cell that normally would give you a tumor. But here, without the possibility to instruct the tissue to have a tumor microenvironment, they're essentially not growing. But there is nothing wrong with this cell if we would now take them out of there and put them in new mice. As long as they can contact the tissue, they can have no problem in forming tumor. So there is no difference between the potential of this cell and the potential of those cells. The only difference is that this cell cannot exploit that potential because the tissue do not allow that the tumor microenvironment formation. So the message here I would like to give you is that even though it's true that a cancer is a disease which starts from a cell, the tumor structure is something that is allowed by the tissue. So it's a disease of the tissue. And I would argue there is even a disease of the whole body in a moment. So we have a situation where a tumor starts. And again, our focus is really in the lab to understand how this special environment is created, how the cells are diverted into the tumor program, even if they're not tumor cells. And then we do that within the primary tumor, but also when cancer cells, which have, of course, the potential that I told you before to restart a new tumor structure, that spread in the circulation, they will do the same thing in different tissue. And the only difference here would be that the tumor microenvironment in the metastasis would be originated by the different tissue where the cancer cell start to grow. And understanding how that process works, again, is a very important question that we are asking in the lab. So metastasis has been one of the main themes of my lab, and particularly how are the early interaction of the tumor cell with the new tissue once the cell have reached that tissue. But one important parameter, and this is that I also would like to share with you, is the fact that tumor is connected from the beginning with the whole body via the vasculature. And this is not only used by the cancer cell to spread the cancer in distant tissue, but is also used even before tumor become metastatic to change the status of the entire body. So the tumor can engage in systemic interaction with the body by releasing a lot of factor and even engaging with the innervation. So one of the measurable changes which happen in people with cancer are changes in the inflammatory cell, and so in the inflammatory cell in the blood, and particularly in neutrophil. And we have a very strong interest in neutrophil because of the study that I'm going to mention in a moment. Neutrophil are a very important immunocell which normally protect the body from the invasion of infection. But they seem to respond very strongly when a primary tumor is there. And this, for example, if I show you here, in the case of breast cancer, we can find that mice that harbors breast cancer, which they're still not metastatic, so they're very small. If we look in their lung, we can see many more neutrophils. So the neutrophil here would be the black dot that you see. This is a normal situation. This is a mouse which harbor a tumor in a distant site. And you see there are many more neutrophils there, and we find more neutrophil also in the blood. Once the tumor cells are spread, now they will seed into the lung and they'll find more neutrophil there. And the neutrophil will engage with the metastasis. So neutrophil is just one of the cellular components of the tumor microenvironment I was telling you about before, particularly at the metastatic site. So when we investigate what this neutrophil do for the tumor cell, we found that they release a factor which is a pro-inflammatory factor, which can normally is talking to other inflammatory cell, but if a cancer cell is around, the cancer cell interject that factor and it profit from it by increasing its level of proliferation. And this is done in a moment where the cancer cells are just infiltrated in the tissue. They still have to start a new growth, and there they go, neutrophil feeding them with a factor which just fosters their growth. And this leukotriene is also important in other inflammatory disease, like, for example, asthma. So there is a particular drug, which is an anti-asthmatic, which specifically blocks the production of this leukotriene. So what we did here is to give this drug, this is the anti-asthmatic, to mice which were having metastatic breast cancer. And here you see a lobe of a lung which is filled with metastasis in a control animal. And when we treat the mice with xylotone, which is this anti-asthmatic that blocks this message, we can already see that there is a lot less metastasis there. And here you can see in a quantification, this is a different model of breast cancer, but still in metastasis to the lung, you have less metastases, which are big. And also when we quantify the colony, now we see that really the colony are much less. So the process is basically stopped from inhibited, let's say, from the beginning. And the thing that is important here to say is that this treatment doesn't touch the cancer cell at all. I mean, this drug doesn't influence the growth of this cancer cell directly at all. The only thing that it does is block one of the advantage that the cancer cell get from the microenvironment. And this is already enough to slow their growth. So in the lab, what we want to achieve is to find those additional value that the microenvironments provide into the tumor cell. And by blocking them, we will make the cancer cell weaker to combine then this treatment with direct targeted therapy, which kill the cancer cell in an attempt to make those treatments more efficient. Now, the last thing that I would like to show you is a strategy that we use to understand a little bit more from what are the fundamental program which drive the rewiring of the microenvironment, the rearrangement of the tissue in the surrounding of metastatic cell. So something we can do, we can use experimental model where cancer cells are labeled with a GFP marker. So this is a green fluorescent protein, which is visible if we hit the cell with a particular wavelength light. So then if we hit the cell with a blue light, we basically get the cell which have this protein to flourish. And here you can see cancer cell growing into a lung. Obviously, only the cancer cell will be visible because they only have their fluorescent protein. But what we are really interested is actually to understand what are the changes in the surrounding of those cells coming from the tissue that allow the cell to grow. So what we have done is to create a strategy where the cancer cell not only has this fluorescent green protein, but also release another protein, which is red, which can be picked up by the neighboring cell. So now the cell is going to tell us who are the tissue cells that they are interacting with, because it will label directly those cells in a red fluorescent way. So now we can see the whole structure, and what we can do, we can make a single cell suspension. So we take the whole tissue and we can investigate all the different compartments. So the cell which have no color would be the cell from the tissue, which is not close to the tumor cell, so it's largely unperturbed. Then there would be the cancer cell, which are now yellow because they have both color. And then there are the red cell, which are the tissue cells. So they're not cancer cell, but they are close to the cancer cell, so they can become red. And then we can ask questions about differences from the normal cell to the cell which now are perturbed by the presence of tumor. And when we did this, I mean, I'm not going to go much in detail. I'm going to share just one of the things that we found, because I thought it was particularly fascinating, because you remember the driving force of the tumor is the ability to read into the whole book of the genome. And normally a tissue cell will have a restriction in the information and will only be able to do one particular thing, which is related to its particular function. But when those cells in the tissue come close to the tumor cell now, they seem to reacquire this embryonic stemness potential, which lead the cell to reacquire more of the information which are available in that book. And this, of course, seems to be a driving program, which, if you can imagine, if the tumor want to rewrite a new structure in their surrounding, that's exactly what you need to do. You need to first wipe what was written before to write something new. So this cell now can do anything that the tumor wants because they are embarking in that program. And the summary that I'll give you here is the fact that the characteristic of cancer will be the genetic cell. The genetic cell will be the tumor cell, which now is gaining this embryonic power, and at the same time is having the ability to rewire the tissue surrounding, gain their special tumor microenvironment, which will be supportive, but also will block the inhibitory effect of the inflammatory cell. Inflammatory cell, actually, in cancer, they're turned more in its favor as opposed to blocking it, in fact. And so just to finish, my definition that I'll give you of cancer is that cancer is a reactivation of an embryonic program of construction. Is true that it's a disease and it ultimately leads to the destruction of the organ, but it is born as a program of construction, which is very much linked to the same program that led to the formation of the whole organism in the first place. It's just that that program is very powerful and is reacquired in the wrong place, so in a developed organ already, and in the wrong time because the organ is already fully formed. So it's not possible for that organ to control this program, and the only thing that is going to happen is that it goes along with it and it embarks into this new program of construction, which is turned out to be ultimately as a program of destruction. And with this, I'll conclude, and I would like to thank the people in my lab that are doing the work, and I'd like to thank you and take a question if there are.